Hi everyone, welcome to this M22 talk. Our speaker today is Alán Aspuru-Guzik. He needs no introduction. So without further ado, let's start.
Okay, thank you, Prudencio. It's very nice to talk to you guys at Valence, and you're doing really great work over there. Before I start, I just want to say my lab is always open to collaborations, interactions, visits, exchanges, whatever. Okay, so if you guys want to reach out to me and there's something that you want to discuss, please go ahead. We're all here in Canada and happy to chat and hang out. Okay, so today I decided in purpose to not give you big AI stuff. I'll tell you more about self-driving labs, and the reason is my AI talks are kind of like 6 months old or something, because I'm from the last AI conference. So I don't have like my newest stuff in slides yet. So I said, you know, I want to tell you guys about self-driving labs. This is my kind of standard talk about it. And I think it's going to be useful because I think they're a great target for AI and a great target for synthesis and characterization of drugs and materials like excipients. Okay, so I just want to begin by talking about the advisor of my advisor. Okay, this guy is called Samuel F. Boyce. Okay, and Samuel F. Boyce in 1955 came to the Boulder, Colorado Quantum Chemistry Conference, unrolled a computer program. This was a quantum chemistry program. And he says, on this side of this tape enters a question. On this side of the tape comes out an answer, right? At that time, it was a Fortran program, right? In 1955. You can imagine what it was, right? Now, you want to read this again and think about what will be the role of AI in the 21st century based on this question, right? This is about almost 80 years after that, right? And here, I think, with the advent of AI, large language models, generative models, etc., that we all know and love, really our goal as theoretical chemists, raise your hand if you're a theoretical or computational chemist here, or okay, put your hand in the Zoom or something. I want to see who's who, right? Our role will be to do that, right? And also, if you're an AI person, raise your hand if you're an AI expert. That will be probably your same goal, to do AI for science, okay? So 5 years ago, I wrote this paper. There were no large language models back then, 6 years ago, right? As good as they are, there was no transformers. But I imagine that this was the future of theoretical chemistry. We're gonna have a dialogue with a robot called Organa that was gonna be making molecules for you. And that is the birth of the self-driving lab, right? So Organa, please make me these molecules that are gonna be good for organic lasers. And Organa was gonna go back and say, yes, Jane. I was very excited back then about very excited about augmented reality, right? So I was thinking maybe everything will be in augmented reality, and maybe with the Apple glasses, it will be. Raise your hand if you have the Apple glasses. I don't, so Valence is not paying you very well, neither the University of Toronto, right? But anyway, I think that's going to come through, right? Now we're going to be talking to AI also with AR, right? And so on, right? So eventually she made the compound. So now let's talk about self-driving labs. What the heck is a self-driving lab, right? So first of all, a self-driving lab is an automated, but also automatic lab. So first, let's talk about automated, okay? Automation, right? I know, sorry, I have this slide first. Sorry. So the main idea of a self-driving lab is to close the loop, right? AI will choose what the next experiments to make. And obviously, this is much more efficient than a linear design, right? And I mean, as you know, in drug discovery, this is kind of like the thesis of it all. I was lucky to write one of the first papers on this topic with experimental validation. You might know my Nature Biotechnology 2019 paper. So 5 years ago, we were the first ones to use AI to generate a drug and test it together with in silico AI, right? And also, we were the first ones to actually do an autoencoder for chemistry in my lab. But I'm not telling you today about autoencoders or generative models too much. I'm going to tell you more about this loop, okay? Because now this is one of the things I care a lot about. Now, generative models, you raise a rock and there's one, right? So now let's think about what's next. So obviously, high fructose experimentation failed in the 80s and 90s, especially for chemistry and materials, because throwing points at random makes no sense. Generative models are fantastic, but usually they generate molecules that are not synthesizable. And obviously, many people, including my group, are working on that. But my idea is to, well, the self-driving lab idea that we employ, right? It's like, let's make a robot that can make a certain amount of chemical space. Tiny. Our robot that I'll tell you about today did 190,000 compounds, can make up 195 compounds out of a chemical space of 10 to the 120 or so potential compounds that you could make if you were an organic chemist. You know those numbers because you're in the field, right? But that's more atoms than the visible universe. But the idea here is that we only run AI on the things that the robot can make or could make easily, right? That way, we actually are searching the valid chemical space. So first, now let's talk about automation. I mean, automation has been huge for chemistry, even bigger for biology, right? So it has been huge for biology and still needed to grow in chemistry, right? And this is an example of a chemist that got the Nobel Prize, right, in 1984 for actually automating the synthesis of peptides, 1966, okay? 10 years before I was born. That's the beginning of automation, very important. But now how about this closed loop idea? Well, I mean, the earliest paper I found was 1978. I am very old, but raise your hand if you have programmed in BASIC. You have never programmed in BASIC, a single line? Okay. Little babies, all of you guys. Oh, one of us. Fantastic. Arthur, great. Give me 5. You're as old as me or something like that. Anyway, so yes, look at this, man. A closed-loop automated chemical synthesis system has been designed for reports optimizing chemical reaction parameters. This system uses a time-sharing computer, sounds like a cloud, right? With the simplex algorithm for optimization programmed in the extended basic language, right? 1976. Pretty much an advanced self-driving lab with a PDP-11 computer. Oh my God, I love this guy, right? So anyway, that's the beginning of the self-driving labs. And obviously now, if you want to learn more about them, we just submitted this paper about 6 months ago to chemical reviews. We just got the reviews back. I think it will get in eventually, right? We have to just go back to reviewers. But this is going to be a 600 reference type paper about self-driving labs. And then this is very important. It's the frugal concept. I believe that the title of this talk is "The Revolution Will Not Be Televised." We're going to be building bottom up all the self-driving labs, right? So this paper is already published in Digital Discovery. So what are the interesting things that we can think in self-driving labs? You want to think about the hardware and how to integrate with it, right? So that's where, I mean, a lot of AI, I mean, I know this is an AI kind of seminar series, but my group and I have worked, for example, in the computer vision issue, on the digital twins issue, also building open hardware from the bottom up and integrating regular hardware. These are the kinds of things that you want to think about if you try to control a lab with AI. And this doesn't have to be expensive. Talking about the frugal self-driving labs, look at this beautiful $300 self-driving lab developed by my former postdoc, Andres, in Mexico, right? So I am a big proponent of democratizing this and taking this to all other countries. And in Mexico, $300 is a big budget, okay? So Andres was able to build this little robot with that. And I think for us, it's very, very, very important that all the self-driving labs actually are democratized to the entire planet, right? But of course, we are in the developed world. So we just got the largest ever grant awarded in Canada. You guys might have heard of IVADO. IVADO has a similar grant, but we're kind of like the peer grant in Toronto. So our grant is only for self-driving labs, right? And we're building a building. If you guys come to Toronto, you will see a hole where this building is going to come up. And the reason I'm saying that is because we need to actually scale for this. We need to think about the operating systems and all the different control systems for this, right? So now let's talk about the organic solid-state lasers, which is the story I want to tell you today about. Raise your hand if you know who is Hendrik Schon. Nobody knows. Okay, good. Hendrik Schon was the biggest fraudster in material science back in the 90s or 2000s. He was working at Bell Labs, and he was building organic superconductors, organic semiconductors, etc. All of them were fake, and all his bosses at Bell Labs didn't see the fake data for a while. Well, I recommend you to read the book. It's very cool. And you can see here that there was an organic laser that suddenly Forrest questioned, if the organic laser was actually good or not. What the heck is an organic laser, right? First of all, well, before that, the postdoc that was trying to verify those results, and we're seeing here Nicole Zhang walking around. It's kind of fun. So you see here, Chihaya Dachi was a postdoc that found the mistake. And he was not happy with that, right? He spent about 20 more years of his life trying to really make an organic laser. And in 2000, what I mean by that is a solid-state emitting organic laser. Why is this such an important technology? Because it's basically what will be powering your AR glasses, okay? Talking about the AR again. It's kind of like the OLED that you see your phone 6 hours a day, but the laser version, okay? The cool thing is that there were only 10 published molecules or so by the time we started working on the self-driving labs. It was an ideal technology to try to accelerate. So to make an organic laser, now this is a little bit like drug discovery, right? You guys care about solubility and Lipinski rules and all sorts of things like that. Toxicity. Here we just care we don't care too much about the shape of the molecule. We care about its energy levels, right? We need to have a molecule that has extremely precise electronic couplings, right? So that it only shuttles the photon back and forth between the excited state and the ground state, and does not have a lot of overlap between the excited and the absorption and emission lines, and a couple other things in physical chemistry that we'll skip. But you can think about it as the analog of drug discovery. You need to actually satisfy all of these different constraints in chemical space. So we got a $12 million grant that we call Madness, and this is a collaboration with Marty, Lee, Bartosz, and Jason. And across the globe, we started working together on actually doing this. And now this is where there's many analogs with drug discovery. We need to define the structure of the molecule, get proxy. This will be, as an example, in vitro type of assays in your language. And then this will be, for example, in vivo type of assay, right? So this is the actual device. You have to make the device. It's kind of harder. You can only make a few devices because you have to ship about 1 gram or 2 grams of material all the way to Japan. And Chihaya makes the devices. We cannot make the devices. But we can see in solution if the molecule is a good laser, okay? So the way we do it is a little bit like in drug discovery when people build libraries. But the difference is that we cannot afford to do the cheap and crappy links that people do, chemical bonds that people do in drug discovery. We need to actually do real chemical bonds. And what I mean by that is click chemistries are cute and they're good for drug discovery, but here we need to make a material, and to make the chemistry click on this material that is very insoluble, we really need to do a lot of chemistry, right? So this is more like hardcore in that sense. So we only, quote unquote, made 1,000 compounds with sweat, compared to, with a DNA encoded library, you can make millions, right? But the compounds that you make in the DNA encoded library are actually way easier to make and way floppier. And they, of course, won't work as a laser, right? So we hooked up a synthesis robot, a HPLC robot, and a characterization optical table, an HPLC machine and a characterization optical table for our Hello World self-driving lab. Synthesis, characterization, I wanted to be able to actually do all of this automated, so we're able to actually make the molecules and all the way to measure them. We can actually measure here the absorption and emission spectra, the temperature fluorescence of the laser, and so on. So Hello World paper came out in 2023 for this type of system. There's a big software work that has derived to many papers to control these things. And we have a chemistry operating system. We have an entire Python library to control this type of robot. We're about to publish a library called Moses for this guy, and so on. So we have developed a lot of open source software to actually release to the world to do self-driving labs. And the first paper, we just wanted to make 40 molecules to start with a single coupling chemistry. This is metaboronic Suzuki coupling. And this is how the system looks like. This is the powder dispenser. This is the synthesizer. And the plots I'm going to show you here are very simple, okay? This is the gain, optical gain. This is the probability that the molecule gives you a photon after you give it a photon, okay? The cross-section of that happening, okay? So the higher, the better. This is a better laser. And then this is the color. So we don't care about which color, but we're going to have the largest possible gain cross-section. And you can see this is Chihaya's best molecule. Even in our first experiment, we already beat Chihaya by a factor of a lot. And actually, the lower this excitation energy threshold, the better the device is. And the narrower this line, the better the laser is. But what matters here is that we made a device that is the best in the world just by starting with the Hello World experiment. So just in this experiment, we already did 4 times more molecules than the 10 that were published before. And we had the best ones. So now let's move on. Now we call this project AC/DC. How can we actually do this at scale? Well, first of all, okay, this is not impressed. The science paper is already out 2 weeks ago. Okay, sorry, I didn't modify the slides. But basically, we just got a paper in Science where we show that to make this thing happen, we have the lab in Japan I told you about, but a lab in Urbana-Champaign, a lab in Toronto, a lab in Vancouver, a lab in Glasgow, and a lab in Poland, all of them working together, making and testing the molecules. The lab in Poland is computational only, but basically, all of the rest of the labs are experimental. Okay? So we had to build a software called Organize It that actually organizes and the logistics of this all, right? Who has which molecule, who made which molecule, where everything is in FedEx, etc. So we literally built a self-driving lab around the planet. Okay. And these are examples of the different self-driving lab robots. We built this platform called Medusa in Toronto. Like if Balance ever wants to make molecules, you guys could also work with us to actually learn how to build these Medusa type platforms. Pharma companies are starting to work with Acceleration Consortium in actually this topic. So feel free to reach out. But here is an example of one of our Medusa platforms in Toronto. And this is the computers at Glasgow and Vancouver, a Medusa platform in Urbana-Champaign. Not Medusa, it's called the machine there. But basically, you can see here all the different types of robots that we control by software remotely. And now, I mean, a little bit of AI, right? How do we do this? Well, I mean, we had to develop an asynchronous Bayesian optimization algorithm, right? That doesn't have batches or anything. As data points come in, it keeps congesting points, and the points actually are masked for a while the experiments are happening. And there's a bunch of details that you can read about in the paper. But definitely, we had to throw a lot of thinking about this, the Bayesian optimization. How many people in the room are organic chemists? I might skip completely the organic chemistry here. Raise your hand if you're an organic chemist. How about if you know organic reactions? Do you know anything about them? Raise your hand. No? Okay. So Prudencio a little bit. So Prudencio, but I'm going to be very quick, okay? We couple, because there's not people interested in that too much, we couple the core to the bridge to the wings using two-step one-pod synthesis, using these fancy protecting groups. I'm going to skip all of that and show you perhaps more our automated platform. We're about to submit this to Nature Protocols. We're able to actually make all sorts of different devices. It's a characterization device. This is an electrochemistry device, a synthesis device. So if you guys want to learn more about Medusa, we're going to release the plans for Medusa open source everywhere so people can build Medusas. This is Medusa in action. You can actually see 95% of the molecules for this project were made with Medusa rather than with the ChemSpeed. And you will see here, I mean, the reaction happened, by, Python control pumps, etc. Along the way, we got another science paper on actually optimizing the reaction conditions for this reaction. This is going to be a huge cottage industry where you can use AI for optimizing generic reaction conditions. So I really like this because remember the first paper in 1976 was about optimizing reactions. Well, here we are about, 40, 50 years later almost, right? Doing the same kind of thing, but now in a science paper for real. So now here's a picture of the organic lasers, about 1,000 of them. We did 500 of them in 3 months. That was the fastest throughput we have. And basically, this basically is the number of powders that we have and delocalized around the planet to make all the compounds for this. We had to develop a little software platform called Organize It that actually allows us to, I'm going to show you the organic lasers, how they're working, see who has what fragment. I'm just showing to you really quickly. Which robots are in which laboratory, and also which fragments are right now being available to synthesize or not. Okay, so here is all the story about some videos about how this system looks like. So now a little bit of machine learning, I guess. How do we predicting an organic laser compound is extremely hard of an issue. Okay. The quantum chemistry for calculating the emission rate of this molecule is almost impossible to get without a very, very large simulation. As a matter of fact, we're collaborating with a group in Vienna to try to see if we can even predict one molecule. So there's no ab initio prediction. We cannot just go ahead and use ab initio prediction directly of this. This is a lot analogous to drug binding. Of course, you can run a free energy perturbation every time and maybe get some numbers, but those numbers might not even be as good compared to experiment. So what we do here is we do 6 DFT properties like excitation energies and things like that that are roughly related to the organic laser, but they are not really the organic laser, right? And we regress against them, okay? And then we use the latent space of that regression model, okay? And to shape our Gaussian process optimization, okay? So in other words, we need to shape the space with the machine learning on similar descriptors to be able to have a space where the Gaussian process can actually optimize well. That's how we did it. Now, the tools for Bayesian optimization of my lab are huge. There's a lot of different developments you can read around here. So we really did a lot of tricks in Bayesian optimization. We put everything together in this paper called Atlas. So a lot of the things that you see here are now condensed in a single software tool. So if you guys want to do Bayes-Opt, you can download Atlas and play with it. Actually, it's a very good library. So we have this multitask Gaussian process regressor. That's how we do it. We have to do it multitask because we're predicting a bunch of different properties. Okay. And something important is that you guys might not have heard too much about ranking-based regression. I don't know, raise your hand if you have ranked regress a drug, but this is going to be maybe the most important lesson for you guys. We found, and you can read the details in this paper, that for material science, and I think also for drug discovery, this will be true. You don't really care about the absolute binding energy. You don't really care about the absolute lasing emission. What you really care is about if a molecule is better than the other. Does that make sense? So predicting the ranking, okay, with this weighted Kendall-Tau formula, that basically this is a Kendall-Tau, and we modified it this way in this paper. So we did a little bit of machine learning research here. This was done by Ben Sanchez-Lengeling, my former student now in Google DeepMind. So he helped us with this idea. He was using this type of ideas for smell prediction. So we don't want to predict what molecule is better absolutely in terms of regression, but we want to predict if it's better than another molecule. And if you do that, the weighted Kendall-Tau, which is the analogous formula to R squared, is 0.91. So we have 0.91 regression. It's not really regression, but it's the 0.91 ability to rank the molecules, you see? And that's what we use for Bayes-Opt. And I think this trick, you can be used for drug discovery, right? You can always say, screw about the absolute regression. I want to just do the ranking. So I recommend you to try that. And here it is, right? We basically spent a lot of time optimizing the synthesis conditions. That's what led to the first science paper. Then we built a 500-point hypercube to actually seed our Bayesian optimizer, right? And then we ran the Bayesian optimizer. And what you see here is the number of materials that were better than the previous material of Chihaya. And so as you can see, we start discovering better and better organic lasers, right? So did the Gaussian process on top of the latent space of the neural network make sense? It does. Look at this cow here. It has like a little cow in its butt. You can see the biggest emitting molecules. So the shaped space actually did help. And this is just a simple UMAP, right? Just to show you that the latent space kind of has some structure to it. And you can see here the devices. I'm very proud of that. Once we made the molecules, we scaled them up, sent them to Japan, and then we made these 3 devices. And these at the moment are the best organic lasers that we know of, right? So this is an example of a success story of AI for materials discovery. Okay, so now it's pretty cool because the Deputy Prime Minister of Canada came to my lab and actually saw the self-driving labs very recently and actually saw the organic laser. You can see here the brightest organic laser in the world. This is a nanomolar concentration. I'm using a laser lamp and it's lasing back to her, right? So that was pretty cool to actually have her visit my lab and announce. She was announcing the new science funding for Canada just right there in between my robots. And I even was wearing a suit, right? I didn't wear a suit to this talk, but I was wearing a suit for that talk. I'm actually wearing a Mexican wrestler t-shirt. Let me show it to you. Look at this cool t-shirt. Anyway, so now let's talk about, for the remaining of this talk, let's talk about the automated lab of the future. And I'm going to leave some time for questions too, okay? So let me argue something with you. Right now, if you look at any automated biology lab or automated chemistry lab, they don't look like the regular lab. Do you guys agree with that? They are full of robots with white machines just moving things, or the liquid dispensers. How many of you have seen one of them? Raise your hand, right? Obviously, you have seen those automated labs. Well, but how many of you have seen a lab at your university or your high school? Do they look the same? No, right? I argue that we have to make humanoid robots enter those labs rather than build automated labs, okay, that look completely differently. And what is that? It is, again, the democratization idea. We are not going to be able to renovate labs in all the countries in the planet, okay? I cannot go to Ecuador and invest $1.5 million in an automated synthesis machine, right? And also, all the chemistry recipes that we know are already made for flasks that we know they're big, made by humans. So the lab of the future, I argue, is going to be humanoid robots using our old-fashioned labs, as well, of course, as these automated new machines. Okay? So I like to take selfies. People followed me when I used to be in Twitter. I like to take a lot of selfies. So we started taking selfies of glassware. Turns out transparent object detection is a very hard problem. And in 2021 and 2022, we had the world record. This is not me. This is Florian Scruti and Animesh Garg, the PIs, and I'm just the chemist in this collaboration. But I am the ones providing kind of the motivation of why to do this and directions and so on. But my grad student, Haoping, learned everything from them. But basically, you can see here, we build a training data set of transparent objects with liquids so that we can use several different sensors to actually combine all the information into our transparent net, our network, to actually detect transparent objects. It's very freaking hard, right? Because light is transmitted and reflected, and therefore most of the sensors get lost, right? Where is this cup, right? You can see, where is it? Is it here, here, and there? Look at this in 2 dimensions. It's even hard for you to know what it is, right? So we also developed this multi-view transparent thing. We got this into one of the machine learning conferences. It's basically when you are drunk and you're trying to open the door of your house, you're probably going to be looking at the door from several angles, try to put the key inside. Kind of, this is the same idea, like the robot actually uses many different views. And now we're using a digital Blender generated data set. So we did a digital data set, and then we use all these different pooling and so on to actually use different frames to actually basically use computer vision machine learning to actually detect where the thing is. We also had to use control algorithms, and this is a very old video of our digital twin trying to be able to pour liquids, right? So this is really cool. I mean, around 2023, this is last year, right? We already were at this level. We were already doing experiments. You know, you can see the camera, so the robot can see, right? So that's very important. But you can see the robot doing here like a recrystallization experiment. So we had to invent this skill, which is a pouring skill. Right? Like we had to learn how to teach robots how to pour things. Very important. And also we're teaching the robots, of course, large language models came out that year as well. Like, I mean, the popular ones. So obviously we also do large language models like everybody else in the universe now. So what we did is try to translate English to chemical description language, which is the language that we use to actually control these robots. But for that, we invented, I mean, many people do it now, obviously, but we were at the time very early as well, like as many people, on the verifier idea, right? So you want to make sure that the large language model has a verification. Obviously, it's not safe or it's not valid. So Clarify is a library that we built to actually do that. And Clarify allows us now then to do experiments like this. You start with natural language, and then you actually do the experiment. So do you guys remember Organa? Okay, now this is the time. Tell me if you can listen remotely well, because I'm going to show you Organa as of 2024. Okay, so my dream of 6 years ago, this is going to be a robot that it's going to do a lot of things. It's going to be interacting with my student Marta, and it's going to be asking her a lot of questions for the planner, a lot of questions about perception, like where are things in the lab, and then it's going to compile, well, basically plan an execution plan and then compile it and then run it. All of this automated. So what you see here, the only thing that we did is we cut the audio, 3 seconds of processing, right? Because it's kind of annoying to wait for the robot for 3 seconds. So look at what happened. Tell me if you can listen to this. Just say something like this. Can you listen?
Hi, my name is Organa. I am a robot chemist. Can I help you with anything today?
Hi, Organa. I would like to run a chemistry experiment.
Could you please specify what the experiment is?
I would like to generate a Torbay plot for an unknown quinone solution.
What is the setup of the lab environment? What are all the reagents and vessels?
So in my lab environment, I have a 10 millimolar solution of an unknown quinone. I have water, I have a 1 molar solution of sodium chloride, and I have 0.5 molar solutions of pH buffers ranging from pH 4 to pH 9. I also have 2 beakers, one experiment beaker and one waste beaker, a potentiostat, and a pH probe.
What is the rationale behind the experiment?
So the goal of the experiment is to measure the potential of the quinone solution at various pHs. We'll start at pH 4 and take a measurement there, and then we'll scan from pH 4 to pH 9.
Could you please provide an example of how to run this experiment?
Yep. So to run this experiment, I would first add 6 milliliters of pH 4 buffer solution. Then I would add 3 milliliters of sodium chloride solution, followed by 15 milliliters of water. Finally, I will add 6 milliliters of the quinone solution. Then I would measure the pH and run a CV scan. And finally, I would transfer the contents from the beaker to the waste beaker.
Can you tell me what the expected outcome of the experiment is?
So I expect that the pH of the solution of the first experiment will be around 4, but a little difference is okay. Now, because it's the first experiment, I don't know what the potential will be, but the general trend is that as the pH goes up, the potential becomes more negative.
What is the number of experiments to be run in total?
6.
Now, you see, it's kind of, I like it because we usually prompt LLMs, but here the LLM is prompting Marta, right? So it's kind of like funny how the LLM is actually giving Marta prompts and Marta is answering them, and then the LLM is learning. It's kind of funny, right? The role is reversed here. Now the computer vision system, the planner needs to know where things are, so otherwise he cannot plan the movement of the arm.
What will this be used for?
That is the washing beaker.
What will this be used?
Now, the cool thing about it is, how did we come up with this? Now we don't use our MB-Trans. We actually use a composition of about 3 or 4 foundational models for vision. I think it's 3 now. Maybe we compounded with ours to make 4, I forget. But basically, this is like a mixture of experts type of thing to actually find the bounding boxes of all these objects.
Used for?
That is the scale.
Okay, thank you. I will start the experiment and let you know if I have any questions.
Now, you can see here, it automatically generates the plots of the experiments. And then I'm fast forwarding a little bit, right? And you can see it here finally generating a report, right? Like any ChatGPT could do or things like that. You actually generate the report, right? But this is a big system, right? Obviously, Organa is not, okay, you can see it there, the GitHub, but I'm trying to jump, wait. Okay, you can see here, of course, you have Clarify as part of it, the perception grounding system. You have the analyzers, you have the task and motion planning, the robot execution software, the perception algorithm, and the actions, right? Remember, I told you that we are teaching all of these different skills to the robot. So pick and place is a very important thing. I have about 3 students this summer working on a better pick and place, right, for chemistry glassware. So what I want to tell you is that this is a very modular system. So Organa can do only those experiments, but we're developing all these modules. And like Medusa, this should be reconfigurable, right? So the idea is that you guys can actually download all this software and start building your own self-driving labs at Balance Labs or whatever, right? So that's really cool. So anyway, here is basically all the different libraries, the robot, the perception library, the NLP library, and you can imagine how they all work with each other. So it's a pretty cool system, right? Now, in terms of time, the robot was 20 times accelerated when you saw that video. It's very slow. So actually, the time it takes for the robot to make the experiment is more than if a human is doing it. You get the same results. But if you look at the NASA task log index, which basically is a questionnaire about how tired you were. The human is very tired doing this crap, right? Whereas the human that talks to Organa can talk to Organa and watch Netflix in the meantime, right? Or play Squad Busters or whatever. So anyway, I asked Marta to give me this because I was like, wow, how does the speech work? So she was using all sorts of this stuff to actually do the speech. This is, I think, already deprecated with the latest version of GPT-4 that speaks like Scarlett Johansson, right? So it's incredible how fast things happen, because I think a lot of this could also have done easier now, I think. But this is how we use the workflow for speech. Just if anybody knows, we use OpenAI Whisper and 11 Labs text-to-voice, etc., etc. So now let me end, okay? And then have questions with a very important question. Does the AI understand science? Raise your hand if you think the AIs understand science. Ihor, you don't believe that? No? Okay. Well, I think you're right? How can AI actually, learn something like a Woodward-Hoffman rule, right? So we wrote this paper basically almost 3 years ago, right, in Nature Reviews Physics, answering that question, but I think a bunch of philosophy questions, right? So we came up with the idea that computer simulation basically has 3 levels, and we only are in levels 1 and 2. They're not levels, or categories, okay? I want you to raise your hand if you have ever used the computer as a computational microscope. In other words, run a molecular dynamics, a quantum chemistry, a stat mix simulation, see the computer do things, and then learn from it, right? That's called a computational microscope. Now, the second one is, and imagine everybody in this room will do that. You do a lot of calculations, right? A lot of numbers, and now you want to see a trend. You inspire yourself and you come up with concepts yourself. How many of you have done that? Right? You haven't done that? Machine learning? How many of you have done machine learning? Right? So that's data science nowadays, right? That's the whole thing. Okay. Sorry, making you raise your hand keeps you awake. Okay. Now, finally, but you have never, unless somebody really believes that, you have never actually seen a computer really learn a concept and tell you about it, right? Maybe you argue that maybe the language models do it. I don't believe that. We're about to put a paper out that believes that's not the case, okay? But we had 2 conditions. They are like Turing tests, okay, in this paper. The Android gained scientific understanding when it can recognize the qualitative consequences of the theory and apply them in a new context, right? This is how we think as humans, right? We learn, we're very few-shot learners, and we can learn something in one context and apply it to another, right? And then the other one is, can you explain it to me? Can you transfer the understanding? So turns out the philosophers believe that if I understand something, I can teach it to you. And it's so true, right? How many of you have taught a class? I think you only understand a subject when you actually teach it, right? If you don't teach a class, you don't understand it. So when I went to Harvard and I taught my first class, I was a professor there for a long time before Trump was elected, and I moved to Canada. In my first class, I was terrified because I was like, yeah, I know quantum mechanics. I really didn't know quantum mechanics, really, I think, as well as until I taught it. So this is a philosophical way of finding out if a robot actually understands. Okay? And with that, I am going to end this talk because I want to have time for questions and answers. I want to thank my research group that is pictured there for all the work that we did. I'm happy to answer any questions that you guys might have. Thank you very much for your time. And I think I have Michael raised his hand. I don't know if it's for a question or he kept it raised. I might stop sharing the slides to be able to talk to you. And then if you don't mind, if you want to ask a question, unlock your camera, tell me who you are, right? What do you do in the company, or where do you work or something? So to meet you, right?
Hi, I'll ask a question.
Yes, who is it?
My name's Aaron Feller. I'm a PhD student at UT Austin.
Great, Aaron. Sorry, I have the grid, so I see you, Aaron. Yes.
Yeah, I'm a machine learning engineer, so working in biological systems, relying on collaborators to validate studies and work that we do. My question is, when do you see this moving to the marketplace or being commercially available?
So there is a company called Chemify, built by my good friend Lee Cronin, which is one of the collaborators I mentioned. He has raised a lot of money, I think 50 million or something. But his system, his idea is that he's not going to give you the self-driving lab. His idea is that he's going to kind of control a big farm of them and then become the Amazon of this thing. I am a democratizer, right? So I have a company called Kebotics that originally wanted to do self-driving labs, but it was too early. So they pivoted to AI for materials discovery. So now my lab is undergoing contracts, well, not my lab, the Acceleration Consortium with a couple of pharmas to give them prototypes. So like, if you guys pay me money, balance, we'll give you a prototype, right? Then with that money, we will bootstrap a startup. And the startup will be based more in an open source type model, a little bit like Python and those guys. You know, it will be a startup that probably will not be as rich, but it's going to be a democratizing type startup. It will sell you things and make money, but its goal will be to democratize, right? And the name of the startup is unknown. But obviously, it will take a while, right? I mean, right now you can buy a ChemSpeed, but the software for it and everything you have to build in-house. And that's why the Acceleration Consortium has a lot of companies like Roche Genentech that work with us because they are building their own self-driving labs. But there are some providers like Excientia and a couple others that actually build you self-driving labs. Arctoris and other companies like that, and in China as well. So in China, I was just in the Insilicos automated lab that was built to them by a Chinese contractor. So pretty impressive. You have seen the crazy videos of Alex Shabarankov's robots. They are true. I just saw them. So it's pretty cool to see the self-driving lab in China. They're I think more advanced than us here in North America, to be honest. Yeah, in terms of self-driving labs built by a third-party commercial vendor. But I'm happy to introduce you to Alex, who can introduce you to his Chinese vendor, and he can build one for you for about $6 million.
A little bit outside my price range right now.
Yeah,
Maybe
well,
one day.
when you become the CEO of Feller Labs, then you can afford it. Yeah. Any other questions?
Yeah, I have a quick question, Alan. At the
Thank
beginning, you were
Prudencio.
saying you have a lot of lab at different places, and I was wondering why that. Is it because the expertise in these places, or just with the intention of democratizing?
No, it's the same thing, Prudencio. It's like we could have gotten the $12 million from DARPA and just built one big lab here in Toronto with everything, right? But what are the disadvantages of that? First of all, there's already equipment and stuff and already things working, self-driving labs in all my collaborators' labs. And we don't have even space. We didn't even at the time have space. Now we're building a building, but we didn't have space to put all of that together in one place, neither the expertise. So we argue in that science paper that the future is going to be like that. Like we have to break the idea of a single lab, a single experiment. It's so constraining. So like the astronomers already think like that, right? Like when they publish a paper, the paper has people from all over the planet, right? So the CERN scientists do work that way. I believe now material science is going to transform into this delocalized thing where by means of software and AI, we can actually coordinate several labs around the planet. And then if a resource is somewhere else, that's great. So I think we're moving into that direction slowly.
Okay, perfect. Thank you. You were saying at some point also that the AI was synthesizing 500 molecules in 3 months. And I was wondering how that compared to having one single chemist working on synthesizing this same 500 molecule in 3 months.
Oh, I love what you're saying. I have the calculation. Let me ask my team. I don't know, but I will argue that making a molecule by hand like that, let's calculate it roughly. It's probably 2 or 3 weeks of work for a single person, right? So maybe say 2 weeks. So we'll have to calculate 500 times 2 weeks. And so I can decide, well, if I wanted to do it in 3 months, how many humans will I need? So let me make that calculation. And then I'll make a picture of a bunch of undergrads, and which I call soft robots. So maybe I will put a picture of a bunch of soft robots and say, I will need so many soft robots to do this.
Cool, thank you. About Organa, do you do something to kind of fine-tune Organa? Like, in for ChatGPT, they do RLHF to fine-tune ChatGPT for different domain and application. Is there something similar you do here for Ghana as well?
No, we haven't fine-tuned. We prompt only. We haven't fine-tuned the large language models for that paper. But obviously, I think Jan LeCun is in Twitter arguing, right? I think I saw him argue that the future is going to be open source language models fine-tuned. We see that. There's a paper. I mean, it's unfortunate. I mean, next, invite me again and I'll tell you more about my machine learning work. But we have a paper that is actually more relevant to what you guys do with Chris Madison, if you guys know him at Vector. He's the main PI on this, and my students worked a lot on it as well, and me. I'm like the second PI, so I was also very involved, but Chris Madison is leading it, where we built a protein sequence predictor that is enriched by text. And this multimodal model, which I can tell you more details once it's published, right? Otherwise, you're going to scoop me well. But it's much better than BLAST at aligning proteins. And what is very nice about it is that when we test it with a bunch of different language models, some of the best ones are actually, fine-tuned chemistry-related models, right? So I think that's what's going to happen. And another project, sorry, this was actually more relevant to another project that we did. Fine-tuned models are better. Let me actually put in the chat a paper that was just accepted in ICML. From our group on that topic. And again, this is more Jeff Pleiss and Agustinus than me. Like, I have many collaborators, but I like to always tell people what I do. I'm the chemist, okay? So, but this was a very cool paper because what we did is we took an LLM. We didn't fine-tune it, but we took an LLM and we removed the next token predictor head and replaced it by a regressor. So we could do a Gaussian process optimization on the latent space of the LLM, and then we could see how well the LLM represents the molecule, right? And a paper that we just submitted to the archive and to the conference, NeurIPS, I guess, is now a Bayesian optimizer, right? An evolutionary optimizer based on similar ideas. And there, this fine-tuned chemistry-oriented language models seem to be really good. There's one very good one from China, from USTC. Happy to send you guys a link to it. It fits in a single GPU, which we also love.
I had a question as well.
Yes, please, go ahead. Who are you?
My name is Farmer. I'm from University of Columbia, graduate student.
University where, sorry?
Of Columbia.
Ah, okay, good.
Yeah, also focused on self-driving labs, but towards biology rather than chemistry. So I was curious, you had mentioned this desire to democratize access to self-driving systems. And some of the ways that you had put forward is bringing these systems into existing labs, either through frugal twins or perhaps eventually through humanoid robots to make use of existing infrastructure. It does seem like another way to democratize as well would be the concept of a cloud lab, a centralized facility that has access to hundreds of instruments, and it could use as its interface something like this chemical descriptor language that you had introduced to submit a protocol, and then it would automatically be executed. What are your thoughts on the balance between Ephemeral Cloud Labs certainly be a prime example. What are your thoughts on the difference between how the research community will be able to contribute to self-driving labs, building AI scientists and connecting them to try to do this kind of objective completion? Do you, between this idea of trying to retrofit an existing lab versus everyone building on a shared platform?
I mean, the biggest problem, I think, for me on the shared platform is the dark reality that for most of the things that we want to do in material science and chemistry, there is not a thing you can buy and integrate, right? So in biology, it's probably more likely because, I mean, what Aleksandr Voronkov did in silico medicine, he grows cells and he does fluorescence on them. And so you can buy Illumina machine for the fluorescence, and you can buy a Hamilton liquid handler, and you can buy everything in 96 well plate, etc., right? Those in robots, like the Mosquito, you know what I'm saying? Like, there's a lot of infrastructure you can just buy and integrate, right? Companies like Thermo Fisher can do that for you as well, right? And I can imagine putting that in the cloud makes some sense. The funny thing about places like Emerald Cloud Labs that they don't tell that to you too much when they advertise it. Is that actually really those labs are humans working with iPads. Have you ever seen their videos? There's a human working with an iPad following the instructions, right? It's a little bit weird, right? It's a little bit kind of like the human is now like this kind of like technician, right? Doing this for you, right? So the question I have for you is ultimately who pays for that technician, right? Obviously, ideally, this could be a public resource, but if it's not a public resource, private money is going to go in. It's not really democratizing anything. So I am very suspicious about this, about the idea of a cloud-based lab as a democratizing agent, unless the government is subsidizing it like a supercomputing center, right? If it's subsidized in a way that anybody can apply to the supercomputing center and just get compute time, right? I can imagine that if you get time in these centralized self-driving labs, then you could imagine democratizing it. Otherwise, you have to have money to do it, and then again, the poor people will be displaced, right? Whereas if I tell people like you can build this $500 self-driving lab yourself in your university in Brazil, right? So somehow the students learn, and it's more of a hacker culture. So I think both will exist, and I'm in favor of both of them, right? I'm just saying they will have slightly more first worldish applications on some of them, and some of the other ones are more educational slash, capacity building. I think we're still in the stage. That's what the title of my talk is: the revolution will not be televised. We're still in the stage that people like you, Chase, that are building self-driving labs in Colombia are hacking there. And we learn from everybody's hacks. So send me your papers, Chase. I don't know what you guys are doing in Colombia. So send me papers so I can cite them in my papers and stuff. So shoot me an email and tell me what you guys are doing in self-driving labs. Yeah. Okay. Any other questions?
I have a question as well, but it's a bit unrelated.
No
Your talk
worries.
is a general question.
Tell me
So
about
I'm
yourself.
Farzana.
Yeah.
I'm a PhD student here at Mila. So I work on tensor networks, and I will be an intern in Zapata soon as well. So I was wondering, what do you think about quantum computing? Like, do you think there will be applications, like it will improve your stack, like somewhere it will fit?
Cool. So yes, some of you might know or not that I also have half of my lab is basically a quantum computing lab, and I usually don't present the two things together, right? But I want to show you one that you guys might find kind of, maybe you might even find controversial. I'm not so sure. But check this out, guys. I wanted to get the archive, but I got this other Harvard thing. Sorry, I don't know why. I want to give you the archive. Well, you can get the archive, okay? Here's the archive. We use quantum computer as a very primitive generative model to generate a sequence that gets transformed to a molecular structure that got transformed to a synthesis. And actually, we attack the KRAS receptor, which is an undruggable receptor. And it's the first quantum computer-generated drug candidate. Pretty crazy freaking project, but because my company, Zapata Computing, which by the way went public, Zapata Computing has generative models in quantum computers. We wanted to see if we use those generative models in the context of drug discovery, we could actually do something. So we're not claiming that the quantum computer is better pretty much at doing this. We're just claiming the quantum computer can do it. So this is really cool. An IBM quantum computer, of course, heavily assisted by classical computers, heavily assisted, was able to generate candidates for drug discovery. So yes, my lab is working a lot in quantum computing. We recently introduced an algorithm called the GQE, the generative quantum eigensolver. I think I encourage you to actually look at this paper. This is probably the best development from my lab in quantum computing this year. So besides the other two papers there, those are the two papers that I encourage you to check out from my lab. But yeah, personally, I'm still doing quantum computing. And when will it help the self-driving labs? Well, I had this idea of doing QA/OA optimization for the robotic planning. And I think a group member of mine is working on that. So yeah.
Cool. Thank you very much.
Perfect. We can take one last question and then we end. Who want to go?
Well, thank you everybody for your time. Who's the last one? As we say in Spanish, nos vamos. Yeah.
I can go.
Okay.
I'm Miruna. I'm a PhD student at the University of Cambridge and an intern in Valence Labs as of the beginning of this week. You mentioned at some point that you see the future of these automated labs as human-sized, and I was curious why, because there's like there would be a huge loss of energy, right, if you make humanoid robots act in these labs rather than box-sized labs.
Because let me tell you something interesting, Miruna, that I found over the years. Organic chemistry. Okay, I'll give you an example. Okay, how many of you like paella here? How many have eaten paella? Do you know the Spanish rice? Risotto. Raise your hand if you like paella or risotto. Okay, so let's just make a paella. Okay, how big is a paella? It's about this size, right? Fantastic dish, right? You can make it vegetarian if anybody's vegetarian or vegan and don't like seafood, right? But let's make the paella smaller and smaller and smaller and smaller and smaller and smaller. How about if I make a paella this size? What do you think about that? Right? Do you think the heat transfer and the liquid transfer and everything of the recipe for making the paella this size will translate to a paella this size? So the knowledge of chemistry, has flaskware about this size, right? So I agree with you. There's a miniaturization direction, like we do already in the biology labs, and also we do in my lab. We want to miniaturize some things and use less material, right? But at the same time, we are forgetting all the knowledge that we have and all the recipes, right, that we have, like making paella, that will have to be translated to make a paella this size, right? And as you know,
Yeah.
fast food is really bad. Like a hamburger made in McDonald's is worse than a hamburger made in a bistro, right? So
no, I agree that scale matters, but then you could have robotic arms rather than the whole body.
Oh yeah,
operating on the same size
of
scale.
course. I mean, why am I thinking robotic arms instead of of? Yes, my friend Andy Cooper has mobile arms. Okay. The anthropomorphic idea comes from the fact, Miruna, that we spend a lot of time designing grippers and pipettes and things like that also that are not compatible with the human hand, because my robot arm is a Universal Robotics or a Franca, and it has very shitty arms. So it cannot really pipette. So we have to publish a paper, actually, I put it in the archive, called Digital Pipette, which is a distraction, right? I had to spend a summer with my student Naruki designing a digital pipette because the arm of this robot cannot handle a pipette that I can buy. So there is an argument, and this is where my friends in Sanctuary AI are doing it, right? That, you know, a lot of the world is already humanized, right? Like a Ziploc bag. I like the example of a Ziploc bag. If you want your robot to open a Ziploc bag, you will need to build a gripper for opening a Ziploc bag. Maybe the Ziploc bag contains your vial for your reaction. So the robot needs to open the Ziploc bag to actually take out the stuff and do the reaction, you see? So if you go down that line, it's actually useful to have robots that can do this. So that's where this comes from. And again, to the argument of my friend that just, I forget his name, but sorry, the student from Colombia that was asking this question, I think he was Chase, right? I mean, the student from Colombia that was asking this question of like, how about cloud labs? They will be cloud labs, they will be miniaturized labs, they will be robotic arms, and they will be humanoid robots. This is not a question of which one, it's a question of let's all develop them all together, right? But the humanoid, I think, will have a big space, a big place, because then you can do any human-based recipe. So I'm moving towards that direction, obviously via the robot arms. We're about to buy our first one. I think they now cost $20,000. You know that? They're smaller, as you were saying. They're about like this size, but we're about to buy our first humanoid robot. I'm so excited. Maybe it's going to be in the corner there just for show, but I want to have it there, right?